Yeah, good morning, everyone. Welcome to Saskatoon. I've got the honour of chairing this first session, which is in relation to the Phenometrics theme within the PERC. And this morning we're going to hear some talks relating to phenomic platforms, so high-throughput phenotyping platforms, both in the field and in controlled environments. So first of all, we have Dr. Allison Thompson, who is from the United States Department of Agriculture in Maricopa. She's with the research services there. And Alison is involved in, she has a cotton breeding program, and she has particular interest in abiotic stress, but she's also in charge of the developing the field phenotyping platform at Maricopa. So without further ado, I'll invite Alison to come up and enlighten us on the program down there.
All right, well, thank you. I'd like to thank the organizers for having me. This is a great opportunity to share what we've been doing in Maricopa. So I'm going to, as Dr. Sharp said, tell you what we've been doing in the arid Southwest in my cotton breeding program. I know you guys don't grow cotton here, but I think what we've been doing could definitely translate to other crops. So tell you a little bit about where I work, the U.S. Arid Land Agricultural Research Center, and what we do within the cotton breeding program and how the high-throughput phenotyping program works with that, and give you a little bit of history of it. Then I'm going to tell you about what I call the older data collection platforms. They were actually developed before I got to ALARC, and I inherited them, and what the target phenotypes for that were and some of the data outputs. Talk a little bit about data processing and management, and then I'm going to tell you about the new platforms and sensors that we've been working on since I've gotten there, and go from there with future directions and some concluding statements. So ALARC is located on the 2,100-acre research extension farm from the University of Arizona in Maricopa, Arizona. And as you can see on the little map here, we are very hot and dry. And it's a wonderful environment to look at water deficit and high heat stress on crops. So all of the breeders in my unit focus on high heat and water deficit. I just so happen to specialize in cotton and how that affects our yield and fiber quality. We have been developing the high throughput phenotyping to help make this process more efficient and effective for the breeders, and to be able to characterize the physiological mechanisms that are driving the tolerance. I'm not going to do an acknowledgement slide at the end, so I just wanted to say most of the research that we do is funded by Cotton Incorporated as well as the US government. So why cotton? It is a $5 billion industry for the United States. The majority of cotton is grown in what we call the cotton belt, which is the southern part of the US. A lot of the cotton is grown over here where it's hot and humid, not so much dry, but the majority of cotton is grown in the panhandle of Texas, which has a very similar environment to Maricopa of being hot and dry. So the breeders from Texas love to send me stuff and say, hey, if it can survive in Arizona, it will make it in Texas. So far that's actually not been true, but we're learning as we go. We're just different enough. So then, getting into the high-throughput phenotyping program, when I started in Maricopa in 2015, I was cleaning out, the old breeders' offices and boxes and stuff, and I found this image, this very first one here. And what they were trying to do was actually take aerial photos of their cotton breeding plots to be able to more efficiently score tolerance to disease. So this concept, I'm sure many of you know, is really not new. Our safety officer would never let us do that now. You can see he's on a very tall ladder mounted onto a flatbed pickup truck that they would drive through the field. And the pictures that they got off of this were great. They would score them in black and white. It worked very well for them. But fast forwarding to 2009, that's when they developed the first high clearance, high throughput phenotyping tractor that you see here in blue. And I believe Pedro Andrade Sanchez was here last year, and so he might have shown you that picture. We have since upgraded to the one that you see here in red, and that was because air conditioning is a wonderful thing in the desert. So those two platforms were already in place when I got there. But then, I was like, I don't really care for the tractors as much, and I'll explain why in a little bit. So I focus primarily on what we call the proximal sensing carts. We've gone through several iterations of those, and that's where I'll spend the majority of my time. I'm not going to talk about it, but I would be remiss not to point out that we do have the TerraRef field scanner. It's one of the largest ones in the world that we have there as well. And we've been working with the drones, and then we have been working with what are termed the robots. So the original tractors focused on what we now call the core trimetrics of phenotypes, and that was canopy temperature with an infrared thermometer, canopy height from ultrasonic transducers, and canopy reflectance. We've tweaked which sensors we use in this particular case. Most recently, it's been crop circles, looking at NDVI biomass. So that's what they started with, and when asked why did they start with that, well, it's because they had experience with these sensors from previous studies, and they were pretty darn sure that they could make them work. So that's the only reason that they started with those. And for proof of concept, they put out a paper in 2014 and showed that yes, you can mount these sensors to a high clearance tractor. Yes, you can collect traits from, in this case, it was cotton. And yes, these traits are repeatable throughout the season, and you can get multiple collections throughout the season. So that being said, they then moved on to the next stage and said, well, are these phenotypes at all useful for genetic mapping? So again, in cotton, they took a small recombinant inbred line population, and they were able to map quantitative trait loci associated with stress-responsive traits. In this particular case, the cotton genome sequence had just been released, and they were actually able to identify putative genes in their QTL regions that actually had something to do with drought stress, ABA responsive, and the carbon isotope discrimination. So that was really exciting to see. So they've shown that this will work. And I think they've done a very good job with that. But again, all that was done before I got there. When I got there, I was handed these two tractors, these papers, and said, hey, this works, have fun. What I quickly learned is what I think most people have been learning. That's a lot of data to slog through, a lot of data to manage. And I am not a computational scientist at all. I've been learning how to code as I go, but I came into it with SAS. That's what I knew, and that was it. So this particular slide, I think Dr. Kelly Thorpe presented this at your very first meeting that you had. So it should be familiar, but this just recaps the process of taking all that data off of the tractor and associating it with our experimental plots through GPS. So all of that's the same. He did have a pseudo processing pipeline in place. He had tools in place to be able to do this for the data, but what I quickly found out is just was not efficient enough. It was taking me too long to get that data associated to my experimental plots to then have it be useful to make a breeding decision in time for next season. That's just not going to work. So the real first thing that I contributed to the program was to say, all right, we're going to try and automate this as much as possible so that I can get my answers much more quickly and be able to keep up with the number of collections that we're doing per year. So with the help of our local IT guy, we brought in some very talented students, Jacob Long, David Muller, and then more recently Chayton Olivieri, who basically took Dr. Thorpe's old program, tore it apart, and put the pieces back together in such a way that it's fully automated for me. All I have to do is open up the application on our server, direct it to the folder that has all of the files from the run that we collected that day, and press go. And it's fabulous. I love it. 10 minutes later, all of my raw data has been uploaded into the database, which I'll talk about in just a second. And then I can go back into the database and query out all the processed data. It's all been geospatially located. All the calculations have been done. All I got to do is pull it out into my program of choice and do my statistical analysis on it. It's fabulous. So as part of what they've been doing, they've developed a couple other applications that just help in the early phases of keeping this data organized. So this run helper just ensures that we have all the appropriate folders and files in place so the application will run correctly. And then each platform that we have has its own application. We've done that to make things easier on the users. Rather than just opening up one big application, you got to click a whole bunch of things and figure out which platform and sensors you want to run. Each one has its own unique application, so it works very well. And each application ties into the database. So this is Mike Roybal, the guy who helped get all of this started. And his son, actually, Brad Roybal. They're both very into database and coding. And then this is Devin Lindsay. They helped develop the database for all of the data that we collect. I told them I wanted everything to be open source, so they chose PostgreSQL as the language for this. And the database is separated into 3 different areas, which are denoted by the function that they perform. So when I open my application, it automatically connects to the database, and the database says, okay, I'm ready, let's go. So the first area then is in the staging area. Once all that data's been shoved over into the database, all those calculations for that geospatial, the algorithms, all of that's happening in that staging area. And then it gets shoved into the production area, which is the final destination for all the processed data. And this area, we cannot touch it with any of our other programs, and that's to make sure that we're keeping the integrity of that raw and processed data safe. Where you can then access all of that data is through the public side, which can be queried through any number of other programs. GIS software if you want to visualize the data, Python for downstream analysis, Jupyter Hubs, which are basically Python notebooks, and then Shiny, our Shiny Studio for further statistical analysis. So they can all access it. All you got to do is give them the database's ID number. So that's great. So that was my first contribution to the program. And then I said, all right, now I want to look into, and this is the overall workflow here, sorry. So all the data is fed onto the server. It launches the virtual machine, which is what houses the database. And then everything else can be done. We do have a swarm of computers. We had to do that because with so many users, we were bogging the system down. So now if you're wanting to do image analysis, it has its own computer cluster for that. And then if you're wanting to do the other types of analysis, we have our own computer cluster for that. So it just helps keep everything running much more smoothly with the more users that we add. So now getting into the new platforms, when I was talking with other breeders, they were very hesitant to try and develop a high clearance tractor. And when asked why, they kind of looked at me like I was an idiot, and they're like, well, we can't afford a high clearance tractor, Allison. It's fair enough. It's fair enough. The other thing that they pointed out is, breeders have plots all over their given area that they're trying to breed for. And high clearance tractors are not easy to move. It requires special permits, at least in the United States, and various other things to be able to move them around. So those were the 2 biggest setbacks. So in response to that, actually, Dr. Jeff White in 2013 developed the first proximal sensing card, again, before I got there. But after I decided, okay, the tractors are going to be lower priority for me to meet the breeder's needs, I took that for a test run, and I absolutely hated it. It was a good first design, but it was cumbersome to push through the field, and because of the locked wheel system, it actually made turning corners very challenging. So we said, all right, what can we do to fix it? And the easiest solution was to just cut it in half. That makes it much more maneuverable. So that was the next design, and I actually presented this at the Transformative Workshop in Ottawa not that long ago. And so we have since published this. You can get great data from these little carts, but the biggest problem that we notice is it takes a lot longer to traverse the field because now I can only capture 1 row at a time. The other thing that we noticed is that temperature increases, so does the pusher's pace. They don't want to be out there in the heat, go figure. So the problem with that, though, is you can kind of see here, it makes the amount of data that you're collecting per plot very variable. And we don't want that. We want that to be much more consistent for the sake of our statistical analysis and our models that we're running. So unless you're willing to be out there with them with a timer and a whip saying mush, keep up the pace, it's not going to work as well. So having the very clever and smart students that I have, they said, well, why don't we motorize these things and make them remote control? So the main students on that were Henry Schrock, Talon Mills, and Alex Conrad, and hopefully this will work. Nope. It should be playing a movie right now. Can you guys hit the button so they'll go? There they go. All right. So they are remote controlled. You can see in the first picture here, we call this Wolverine. It has all the carts are named after Marvel's comic characters, in case you're wondering about that. They have a remote control in their hand, and they flip a switch and it has cruise control. So all they have to do is make sure that it drives in a straight line. So we have two of these motorized carts. Wolverine focuses on the core trimetric, the same as the tractor. It was meant to be the replacement for the high clearance tractor, so to speak. And then we have the Professor, which is our imaging cart. So this has greatly fixed the problems with the initial carts that we have had. And they've been working very well for us. So Wolverine, to claim that it's a true comparison to the high clearance tractors, we did a head-to-head comparison with it. The cart actually is able to finish the field before the tractor because the cart can do a 360 turn, whereas the tractor cannot. So on the tight corners of traversing through the field, the cart does a much better job, and you can actually shave half an hour off of your collection time, which is great. Comparing the sensors, we had good correlation with height output between the two platforms using the same sensors. Now it's a little hard to see, but the root mean square error was 5.1 centimeters difference, and the correlation was 0.937. The difference in the sensors is likely the sensor-to-sensor variation. If you read the manufacturer specs, the height at which we have the sensors mounted, their resolution is 5.3, so we're well within that. That's just the natural variation of the sensors, not any problem with the cart itself. We did have a lower correlation with canopy temperature, but that was not unexpected. The tractor fell behind by as much as 2 minutes compared to the cart, and so canopy temperature is a very dynamic trait. It changes very rapidly in 2 minutes, but the fact that we still got a positive correlation was good to see. So they're very comparable. You'll notice I'm not talking about the canopy spectroreflectance, and I'll tell you why at the tail end. So then getting into the Professor cart, the imaging cart, we developed a processing pipeline for that initially before I had the team of wonderful students that I do. And we developed a workflow that worked okay. They have since improved it. But we've gone after a couple different traits with the images. I'm going to focus on what I call the bowl isolation trait. It's not really counting the bowls, but it's just isolating them. And we do that for yield estimate. If you have 10,000 F2, F3, or F3, F4 plots in the field, and you have to hand pick them because they're too small to pick with the picker, you don't want to have to pick them all. So the idea is, can we figure out which ones are going to be the best and leave the duds behind without actually harvesting them? So with that, we were able to develop plot-level orthomosaics that you can then threshold the bowls out and basically just do a pixel analysis and from the cart. And with that, you get an R-square value of 0.67, which is not bad. Unfortunately, it was not significant, but I think that's because the sample number in this particular year was so low. It took a long time. We have since improved that, and this year I'm hoping we'll get much better. The reason I'm focusing on the bowl isolation trait for this particular platform is we've been focused on it with other sensors as well. This is a very popular trait that the breeders want. So the other thing that we've looked into is, can we use light detection and ranging, or LiDAR, to be able to actually count the bowls, because you're getting that 3D cloud. And then we also wanted to know, can we locate the bowl along the plant? Because that gives us an estimate of quality. In cotton, the best quality bowls tend to be in the middle of the plant, as opposed to the top or the bottom. So we had 2 LiDAR units. We mounted one to the tractor, and we mounted one to the professor cart, and took them through the field. And if you're wondering why we have colored cotton, that was part of the validation ground truthing that we did for these particular traits. So you can get a very nice 3D cloud of cotton. It doesn't necessarily look like cotton. There's a video on this one too, if you could click the button. But basically you can successfully count bowl number, and you can get height out of this. But the data processing is incredibly time consuming. Even with our amazing students, this still takes a very long time. It's just not quite as efficient as I would like. Part of the reason it takes so long is the cluster thresholding process. It's very dynamic. If you set it too loosely, one large bowl might actually be counted as 2 bowls. And if you set it too tightly, 2 bowls that are close to each other might only be considered 1 bowl. That's a problem. And basically, the only reason you know that's a bowl and rather just a cluster of dots is because I'm telling you it's a bowl. So you don't have good textural consistency in these models, but you do get that with images. You get great textural consistency. You can tell it's a bowl, but you don't get the restructured geometry to go with it. There's another video in this one. So with the Professor platform, we actually have 3 cameras mounted on it in a triangular array that you can then develop 3D point clouds with the imagery from, as well as those orthomosaics. So the idea with that then is to drape the LiDAR data on top of the RGB data to get the geometry that you want, as well as the textural continuity. And this is an example that it can be done. Again, very computationally intensive, so we're kind of rethinking about how we're going to utilize this technology the best. But it can be done. So then, again, continuing the bowl isolation idea, we have been working with the drones quite a bit. And so we wanted to see if maybe the drones would be a more efficient way of getting this after bowl isolation, because the cart still takes a little while to traverse the field and do all that analysis. So this was just a simple drone that we flew with an RGB camera. The processing pipeline is very similar to what we have with the cart, and you get these beautiful orthomosaics that you then threshold the bowls out. And what you'll notice here is we actually had a lower correlation with yield than we did with the carts. That could simply be resolution, but at least it was significant, but that's because this process is much faster than the cart. The other thing that we noticed, though, as we were going through this, is plant architecture for cotton is not ideal for a nadir view, so this top-down view. And that's because these top bowls occlude the bottom bowls, and so you're missing them regardless with a drone or with a cart, even if the cart resolution's better, if you're only getting that 2D orthomosaic. So if we really want to get after this, we're going to have to continue in that 3D space to be able to see all of the bowls. The other problem, though, that we're running into is bowl density. And you can see over here in these pictures down below, these bowls are roughly the same size based off of the grid behind them. But it might be a little hard to see. You can see this bowl here. It looks lighter and fluffier, almost like a cotton ball that you would get at the grocery store. Whereas this one here, the cotton is darker and much more tightly compact, and that's because it's much denser. There's more cotton packed into those locks of that bowl than the other one. So it's going to yield better than that one will. And I don't know how we can get after that particular phenotype with what we have so far. if anybody has any ideas, I would love to hear it, because that's a very big factor of yielding cotton. The other thing that we've been working on with the drones is fractional vegetation cover. In this particular study, we used the spectral reflectance cameras, but we have since found that it works with the RGB cameras just as well. But we were going after the fractional vegetation cover for a soil water balance model. And that's a lot of engineering math that I'm not going to pretend to fully understand. But at the end of the day, what Dr. Kelly Thorpe was able to do was to estimate evapotranspiration from his models. And then, because he had all this data over time, weather and the fractional vegetation cover, he was actually able to parse out the transpiration of evapotranspiration for each of my plots. And because of that, we were able to calculate total water use efficiency for my plants. So we were very excited by this. This was one of our first good outcomes of a modeled phenotype. That worked very well. So we were actually able to identify 2 cultivars, 1 and 4, that had pretty consistent total water use efficiency across 2 planting dates, so trying to get after the high heat, as well as across 4 different watering schemes, 60% all the way up to 120% of what the water should have been for that cotton crop. And so that's good. I like those lines. They're consistent regardless of what environment I put them in. So that's good for the producers. They know it will do well. So that paper is going to be coming out soon. It's in revision at the moment. So then getting into the last platform that we've been working on are these so-called robots. There's another video here. These became popular with really tall row crops because you can't easily get a tractor or a cart over these particular crops. So they decided, well, we're just going to go in between them and have this mast that has all these sensors mounted to it that look into the canopy. Obviously, we don't need that for cotton, but a group of breeders approached me and said, hey, we want to be able to look at light penetration through the canopy. We think that the different canopy architectures of cotton, typically a Christmas tree, but it can also be columnar or an inverted Christmas tree. We're wondering if one of those architectural types is better for yield and or quality, but we want to see the light penetration coming through the canopy. So we bought this cheap little platform here. Its name is Cyclops, if you were curious, because it only does 1 row. It's $150 off of Amazon, and Alex Manning put it together, put the mass on it with 2 photosynthetically active radiation sensors, 1 above and 1 below, and lo and behold, we are able to capture percent light penetration through the canopy. Whether that's useful for breeding is yet to be seen. We just wanted to see if we could do it, and we could. We also decided that while we're down there, we're going to throw a camera on this thing, because as I said before, a nadir view is not really conducive for cotton. So we're like, hey, we're underneath it now. Let's look up into the canopy and see if we can get some better results of what's going on. So we still need to work out some of the placement of that camera, but as you saw in the video, you get a very good understanding of the architecture from underneath. And then you'll also notice in that video, he's not holding a remote control. He made it fully autonomous, and the reason that we did that is clearly to be able to run this through a field, you're going to have a lot of them out there, or it's going to take all day. So the idea was to create a little swarm of these guys just circumnavigating the field on their own. And then we'll also take that technology and move it onto our carts as well. So now all the students have to do is sit in the air-conditioned pickup truck and watch them go. They're very excited by the idea. So we said proof of concept done with this, and we're moving on to the next phase. So we bought a much more robust platform. That one's about $3,000, again, off of Amazon. You can get everything from Amazon. He's working on getting that put together now, and we're happy to move on to the next phase. Oops, there it goes. So some future directions, I purposely left out canopy reflectance earlier. I have not been real happy with this particular metric for our location. It could be the sensors, it could be how we're trying to capture it, or it could just simply be that the wavelengths that have worked everywhere else just don't work in very, very sunny and hot Arizona. So we've got some new experiments coming up to try and look at that, some different sensors, how we're capturing it, and the wavelengths. We're continuing to work on the bull location with that LiDAR and RGB cloud data. We're going after stand counts, germination efficiency. We're doing that with the drones and with the Professor cart. And then photosynthesis. We were actually able to purchase what's called a laser-induced fluorescence transient system. The original paper on that by Colbert is down below. And we actually had some collaborators come last winter to Arizona and run this system on one of the carts over some durum wheat, and they got some really great results, and they're actually going to be doing some genetic mapping studies with that data. We said, well, all right, let's get one of these, we'll put it on a cart and see how it does in an Arizona summer. So that remains to be seen. But it's a very good sensor. We like it. The group that produces it has been very open about how it works, and so that's really important when considering a new sensor that you're going to work with. So some conclusions. We do have a data processing and management workflow that we like really well. All of the platforms are incorporated into that workflow now and incorporated into the database. The high clearance tractors work well, but are cost effective and prohibitive for, not cost effective, cost prohibitive for many programs. Need more coffee, I guess. The proximal sensing carts are very efficient alternative. I like them very well. And we're continuing to make improvements on them. There's still some tweaks that need to be worked out. The drones, we've been pretty happy with them so far. They're continuing to advance, and so we're still exploring all the different traits that we can capture with the drones. And then for the phenotypes, the ones that we're fairly happy, well, I love canopy temperature. That's a great selection criteria for cotton in Arizona. But we're very happy with that one. Canopy height, we are exploring new methods of calculating that. We're looking with the drones as well. And we actually have a paper on that coming out soon. Canopy cover works well with the spectral cameras or with RGB cameras, and you can get a lot out of that, particularly that total water use efficiency. Bull isolation and number, we're continuing to work on it, but it does work. And then the light penetration, we have that proof of concept made, and so it's just a matter of determining if it's really as useful for the breeding program as the other breeders think it will be.